Now, we've been talking about molecular cloning and looking at genes of organisms. There's another way of looking at the way biological systems and cells and organisms work, and that is to look at them holistically, studying organisms on a grand scale, which brings us to the topic of genomics and proteomics. So the genomics basically is a study of the entire genome of an organism. Rather than looking at individual genes one by one, you look at a complete set of genes and their nucleotide sequence, how they're organized, and how these arrangement of organisms change over time, how do they compare to the genome of other species. Proteomics is in contrast to studying the proteins inside cells one at a time. This is when you look at the entire complement of proteins that is produced in a cell. So basically, you can look at everything in a grand scale and, let's say, compare a cancer cell with a normal cell in terms of the entire pattern of proteins that it produces. Now, genome sequencing has allowed us to do a lot of genomics on organisms. Right now, sequencing is quite rapid, and scientists are able to look at the entire genome of an organism for a very short time. And sequencing the entire genome of organism has really helped advance our studying of life. So these are some of the organisms whose genome were first identified, like mouse, Drosophila, C. elegans, Saccharomyces, and then plants. But as the sequencing becomes more and more efficient, we can much more rapidly get an entire genome of the organisms and study them. Now, an interesting finding of looking at the genome of human beings, and when we did the Human Genome Project, was that among the billions of base pairs that a human cell has, only a small fraction of it is used to make protein. So this is how the entire genome of the organism of human beings are divided in terms of how they're classified in terms of their function. And you can see that only 2% of our genome is used to make protein. And there's a whole lot of sequence that it's really their function is not entirely clear, and it in the beginning, scientists used to call them junk DNA, but right now we are learning that, for example, introns play a very important role in regulation, and we are slowly understanding the function of the rest of the protein. So as a scientist, when you look at the sequence of the genome of an organism and ask yourself, where this sequence that I'm looking at, does it really code for a protein? And that's where proteomics can come in, and you can verify the conclusions you made from looking at the sequence at a genome level, and really verify whether the sequence that you have been studying is really coding for a protein. So at the end, I have included a couple of images for you to practice your understanding of the material. This is from the CRISPR-Cas9, and this is an exercise where you draw the steps in cloning a gene. We're assuming that it's the human growth hormone. This is the restriction site. So draw the steps of cloning this human growth hormone into this plasmid, as you learned in this lesson.